Well, hello, ladies and gentlemen. Hope you're doing well today. So, eventually, we're going to be getting into the body systems and looking at what the systems do, but we're still sort of building up to that. Chapter 3 is sort of a catch all chapter that looks at a lot of different topics. So, we're going to hit on several different topics in Chapter 3, leading us ultimately into tissues. And once we get to tissues, then in Chapter 4, we get into organs and organ systems. So, again, we're still sort of building up to our understanding of organ systems. So this will be a multi-part lecture with lots of little clips over different specialized topics. So let's first look at organelles, the little organs that operate inside of cells. The first structure we'll look at is the nucleus. Nucleus is, of course, important because it contains genetic material, the DNA. Of course, it's wrapped around that histone protein, and that's what we refer to as chromosomes. When the DNA coils up, then it's sort of packaged and ready to ship out, but it's all still the same genetic information. It's got a code that dictates what proteins to build to run the cell. Now, the plasma membrane is a key feature of cells. The membrane is made of phospholipids, arranged in a bilayer, of course, and because they have a nonpolar middle portion, they can sort of keep out certain structures. Anything that's big or charged or polar is going to have trouble fitting through that membrane, so it helps control what enters and leaves the cell. So that's an important feature of membranes, is that they can, again, allow certain substances in and out of the cell and provide the cell with some flexibility. Mitochondria, I know there's a nickname people frequently use for them. I think it's a bit of an undersimplification of what they do, but the basic idea is energy conversion. We eat foods like carbs that have energy, and those molecules can be broken down inside the mitochondrion, and that energy gained from that can be used to make ATP. ATP is sort of the currency that our cells run on, and so mitochondria do an important job converting the energy from our food into the ATP to power the activities of our body. Ribosomes are, a lot of times people draw them as like little dots. Ribosomes, you can think of them as like little factories that assemble proteins. Amino acids are the building blocks of proteins, so you can eat food like tofu or whatever the case may be, and it has proteins in it. Through hydrolysis reactions, we break down those proteins into amino acids and send them to our cells. Under the direction of the DNA, then we can assemble those amino acids into the proteins, and when the proteins fold up, they do different jobs, and they run our bodies. The rough ER is one place where those proteins can be made. Proteins that will ultimately be exported from the cell are initially made on ribosomes in the rough ER. The rough ER is rough because it's studded with those little ribosomes. So in this image, you can see how you start to make a protein, and it gets packaged up in the rough ER and sent on to the Golgi for later processing. We'll talk about that more later. Another membranous structure that we see is the smooth ER. The smooth ER is smooth because it doesn't have ribosomes. And because it doesn't have ribosomes, it performs different functions. The smooth ER is membranous with those sort of membrane-like pouches, and what it does is it actually makes membranes or other nonpolar substances. It also contains enzymes inside of the little pouches to help detoxify different molecules. The Golgi receives materials made by the rough ER and activates those proteins and then ships them out to the appropriate location. Sometimes proteins that are made in the rough ER need to be used in the cell. Sometimes those proteins need to be shipped out of the cell. What the Golgi does is contain enzymes that activate those proteins and then send them to the appropriate location. Lysosomes. Lyse means to split. So lysosomes have hydrolytic enzymes in them that cut up old cell parts. Or sometimes if we have like a macrophage, which is like an immune system cell, it will capture pathogens from outside the body and destroy them. So these enzymes are hydrolytic. They break the bonds between atoms in the molecules, and then you can, again, reuse old cell parts. Peroxisomes. Peroxisomes break down things like hydrogen peroxide. Hydrogen peroxide is really reactive, and it's damaging the tissues in the body. But hydrogen peroxide is produced by chemical reactions in our body. So we produce this toxic substance, so we got to get it out of there. So what peroxisomes have is an enzyme called peroxidase that breaks down hydrogen peroxide and converts it to water and oxygen. So peroxisomes are a way to sort of break down substances that could be harmful to the body. The cytoskeleton is made of proteins. The cytoskeleton, it's like the little lines you draw going here and there in the cell. They help give the cell its shape, and they help sort of anchor structures during mitosis and cytokinesis. So again, they give the cell its shape, but then they help with mitosis. Let's see, some of these cellular extensions. These are the things we can find on the surfaces of cells. Cilia, like we see in the upper right-hand corner here, they're little hair-like projections, and cells will have a lot of them. Now, these are cilia from your respiratory tract, your lungs. So when you breathe in, you're breathing in all sorts of junk, right? And we got to get it out of there. So the cilia sweep that mucus up to the back of your throat so you can spit it out or swallow it, okay? Flagella are long tail-like extensions, and usually in our bodies, they just have one flagellated cell, and that's the sperm. So it produces cell motility. And then microvilli, oh man, the name is appropriate. Microvilli are little tiny hair-like projections on villi, which are sort of like finger-like projections. Villi are like short cilia. So we think of flagellum as long, cilia are short, villi are even tinier. And then microvilli are like tiny hairs on top of that. Well, what happens is all these microvilli create a lot of surface area. So, like the lining of your small intestines, where you absorb all your nutrients and a lot of water, that tissue is lined with microvilli to try and increase the absorptive surface area. All right, I think that ends. Yeah, that ends our discussion of the organelles. So you can take a moment to review those, and we'll go from there. Thank you much, guys. Just got to figure out how to stop this. And here we are. Sorry.